Hey, welcome to the introductory video for summer internship in microbial DNA barcoding. So what is this internship about? It is a 3 weeks intensive training program in analyzing the DNA sequences of microorganisms. When I say microorganism, they encompasses microparasites, yeast, molds, under fungi. You can give us cyanobacteria, actinobacteria, proteobacteria, firmicutes. We are going to include all kinds of bacteria in this internship. So who can participate? All UG students, PG students, especially in their final years planning their thesis. PhD scholars who are planning to have a separate chapter of molecular taxonomy in their thesis. Postdocs and faculty who require species level identification using molecular data. Even enthusiastic school teachers who wants to continue their publication while teaching in the schools or in POCs can still take up this internship because we are going to get two free DNA sequencing from this internship, which cost around 8,000 rupees when you are sending live cultures. Okay, so this internship is actually giving you more than what is going to charge you. So how this works for free sequencing? Any two cultures of bacteria you can send and get it sequenced, or one culture of bacteria, two genes you can ask us to sequence. We will. There is no primary judges separately. You can send us a mixed culture and point out which bacterial colony you want. We will sequence it, pure culture it. We'll give you back with the glycerol stocks and any environmental sample, food, water, anything. The environmental samples are accepted. We will seal, dilute, and plate it. We will find the CFUs and we will give back the actual pure colonies. We will also sequence it to colonies. You can sequence it for free. And if you send us a plant samples, we can also isolate endophyte epiphytes. We can sequence two samples, two microbial strains from that for free. And we will also make a pure culture glycerol stock and send back the glycerol stock with your sequences. If you want to identify a plant infested fungi, if you want to diagnose the species name, just send us the infested part. We will grow the fungi. We will go for DNA extraction and we will tell you what species it is. If you are very much interested in animal samples, you can send us a gut sample or any animal sample that we will isolate bacteria and other molds from it. We can sequence two microorganisms from it. So all those things are done for free. Okay. So the idea behind this free sequencing is to use your own samples to learn DNA sequence analysis. So what are we analyzing? Even UG students who are in the final years of their degree can have a head start because you just have only 3 to 4 months for your project. Imagine you having an characterized identified bacterial species which we can use it for application purposes. Okay, those little time you can have a head start by sequencing through this internship. This internship is also organized outside your academic hour. You don't need to spend time. It is between 7 p.m. and 8 p.m. Even if you miss the class between 7 and 8 every day, the pre-recorded session are actually shared in the group after the live session. You can make use of it. PG students who are especially can plan to have a separate chapter, a molecular biotaxonomy chapter in their thesis, if they are actually, get those genes sequenced through this internship. Also, PhD scholars who are writing a thesis or who has a bacteria only, they may have done till biochemical, they may know only the genus name. We can do the species identification through 6NF RNA sequence. You can send a sample, two sample we are sequencing out of free. So you can be a part of this internship. And more importantly, you can analyze your machine output files, your blast analysis, everything from your own bacteria. You can learn DNA sequence analysis with your own DNA samples. And for faculty members, they can still, either you or your students can take up this internship so that you have your bacterial samples sequenced and your students are actually trained in DNA sequence analysis. This will save most of your time in teaching them and also students will get benefited. If you're forming any new team in your research area, this is an ideal internship to encourage, either you can take it or encourage your students to take it so that the team will be immensely benefited because you will publish the DNA sequence data in NCBI, all two data, and you may use it for writing the manuscript. This is not only particular to microbiologists, anyone interested in veterinary science, forestry, biotechnology, zoology, botany, environmental science, agriculture, pharmacy, anyone interested can take up this internship. And these are the softwares you'll be learning: ChromaSlides, Mega, and you'll be learning ClustalX, and you'll be learning a BioEdit. in Mega, there are a lot of things you will be learning. You'll be learning multiple sequence alignment, phylogenetic tree construction, bootstrap analysis, how to calculate evolutionary distance estimation using p-distance, Jukes counter, and Kimura parameter, and how to do the codon-based testing for selection, Z-test, and model selection. You'll be studying about DNA protein substitution model, base composition statistics, and many more polymorphic analysis can be done using Mega. So this internship is basically to take your sample and do a free sequencing and teach you with your own data how to analyze it. I hope this will be useful for UG/PG students, PhD scholars, postdocs, who are dedicatedly planning a molecular taxonomy work as a part of their ongoing work research, and faculty who is ready to form a new team. So I wish you can make use of this internship and get benefited.